Okay, welcome to week 10. We're almost done in the home stretch here. A couple of things I'd like to talk about before we move on to the new material. This week you do have exam 3. I highly recommend that you watch all of this, the beginning and the end, before taking your exam 3. I do have a lot of wonderful poll questions that review, and a lot of the information is found on the exam 3. So that would be in your best interest. I did put a scrolling announcement on your course announcement as well, so that everybody is aware of that. Another thing is your paper is due by Sunday this week. After that, it's 10% per day. And your paper is worth quite a bit of your percentage in the class. So if you're kind of hanging on the brink between an A and a B, or between a B and a C, or passing and failing, that paper can carry you up to the next grade level quite easily. So that's something I recommend you spend some time on. It needs to be APA. It's about point of care testing. You guys were given the information on the paper during weeks 1 or 2 lab, so you should have that with you. If you don't have the information that you need, the rubric for the paper, and you can't find it, make sure you get that from me before you hand it in. The last thing that I want to talk about is on Saturday at noon, I am doing a final exam review. I am going to cover everything quite quickly that we have covered in this course. Everything that is on the exam will be talked about in some way, shape, or form on Saturday. I will also have a wonderful set of poll questions at the end. I know some people mentioned that they had anatomy and physiology lab at noon on some campus, and that's okay. Anatomy and physiology labs generally don't take that long to perform, so I would do the lab at the learning center or the library, something like that, and grab some headphones and get in to at least get the tail end of it and the poll questions at the end. While I know you can go back and watch the archive, I think that being there and participating in the poll questions makes it stick in your brain a little better. It makes you think about those questions. That way, if you do get a question wrong, you know you're not going to get it wrong again in the future. So watching the archive is okay, but definitely being there live is in your best interest. So just wanted to throw that out there. All right. Today we're going to move on to hemoglobin and porphyrins. None of the hemoglobin and porphyrin material is on your exam 3, but it will be on your final exam. There will be about 3 questions from this week's lecture on your final. So just want to be aware, really watch and pay attention as far as where I point out what the important things are. Before we get started with that, we're going to go back and do a little bit of review and some poll questions on last week's material. So hopefully you've been studying up on your blood gases. And if you're still a little leery on blood gases, your lab adjunct is going to do some blood gases with you this week as well. Okay, first question. Which cation is in the greatest intracellular concentration in the bloodstream? The one that is found inside your red cells. That is correct. The answer is potassium. Remember, if you have a potassium that's above the reference range of about 5.5, and it gets really high up in the 6.0, 7.0 range especially, you would not want to report that patient's result. If you get a result like that on the analyzer, you better go back and take that specimen and look at it and check for hemolysis. Usually if someone is that high where they're 6 point something, it is almost lethal for them to have a potassium that high. So the first thing that you would do before reporting that to the doctor is look at the sample, see if it's hemolyzed. If it's not hemolyzed, run it one more time to double check. Because all those red cells burst and leach all that potassium into the blood. Which cation is in the greatest extracellular concentration or outside of the red cell? Yep, the answer is sodium. Sodium is found outside of the cells in a higher concentration than inside. Okay, what anion, meaning negatively charged, all 3 of these here are negatively charged, has the greatest extracellular concentration? And that is correct. The answer is chloride. Chloride and sodium kind of work together to keep a balance because one's positively charged and one's negatively charged. What is the major function of electrolytes within the body? Where the major function of the electrolytes is the maintenance of osmosis and water distribution. What is a buffer and what is the major buffer of the plasma? I'll give you a hint. It was one of the negatively charged cations. Okay, a buffer is something that resists the change in pH. So maintaining pH is a good answer. It can slow down a chemical reaction by resisting the change in pH. So I guess I would slightly accept both of those. But resists a change in pH is what a buffer does. The major buffer of the plasma is bicarb. So the HCO3 was correct. Which of the following could cause respiratory distress? Or acidosis, sorry. Which would be respiratory distress? Yep, the answer is hypoventilation. When you're not breathing because you've been in an accident, you've been knocked unconscious, or if you have emphysema is another good one, where you're not breathing and you're unable to do appropriate gas exchange, the CO2, which is very acidic, builds up in your bloodstream because you can't exhale that effectively. So it keeps building up and building up. The CO2 comes from the respiration of the cells and things like that. So once that builds up and builds up in your bloodstream, it causes your pH to decrease. Someone with tetany would exhibit which of the following results? A low potassium, a high potassium, a low magnesium, or a high magnesium? The answer for this one is a low magnesium. A low magnesium will cause muscles to tense up and kind of seize. The tetany and the word tetanus kind of go together. You get tetanus, you need a tetanus shot for a certain organism, but tetany refers to what happens when someone has tetanus. Your muscles seize up. So same thing would happen with a low magnesium. Another thing that can cause can cause tetany is a low calcium. So both of those can cause tetany. Someone with an abundance of a heme precursor due to the absence, oh shoot, we didn't get to this yet. The answer for this one is a porphyria. We'll get to that later. Let's get through that. All right, what is the reference range for phosphorus? I'm really tricky on this one. Okay, yes, the answer for this is 2.5 to 5.0. What is the reference range for chloride? The answer for this one is 90 to 109 millimoles per liter. All right, we got some blood gases here. Determine the acid-base status. If the pH is 7.24, the CO2 is 44, and the bicarb is 18. Now, yes, for your final exam, you do have to know your reference ranges for those. And the answers could be metabolic acidosis, metabolic alkalosis, or respiratory alkalosis. Okay, the answer for this one is metabolic acidosis. And for the person who said they're confused with this, I guarantee you you're going to go through 100 of these in lab, so that's going to help you out tremendously this week. I gave tons and tons of sample problems with the answers and why they are those answers to the laboratory instructors. So you're going to go through your lab this week really quickly, which is just a spectrophotometer that you guys are probably getting sick of, and then you're going to spend a lot of time reviewing these blood gases because either you get it or you don't. So I'm not surprised by the second answer on there, and definitely you are not alone. So we are going to go through a lot of these this week, but let me explain the metabolic acidosis. Before you determine your gases, the first thing that you look at is the pH. Is it increased or is it decreased? If it's decreased, it's acidosis. If it's increased, it's alkalosis. A normal pH is 7.35 to 7.45. So this patient having a pH of 7.24 means that it's decreased, so they have an acidosis. The next thing that you need to look at is the CO2 and the bicarb. Because the pH is low and the bicarb is low, the CO2 is slightly high, but we'll get to that in a minute. It would lead us to believe it is a metabolic acidosis. If the pH and bicarb are both low, that would mean we have a metabolic situation. Okay? The CO2 is not that high where the bicarb is very low. So we know that the person has suffered a metabolic acidosis and is starting to recover from that. Okay, here's a tricky one. Determine the acid-base status if the pH is 7.39, the CO2 is 25, and the bicarb is 15. In this case, it's weird because the pH is normal, but the other stuff is abnormal. Go ahead and throw a guess out there, that's okay. Okay, the answer is a compensated metabolic acidosis. Now with this one, the pH is normal, but everything else is abnormal. That's what leads you to put the compensation in front of it. Now because the pH is the mean for the pH where it should be normal, 7.4, because 7.35 to 7.45 is normal, you can tell that it used to be an acidosis state because it's still slightly low, but it's coming back to a 7.4. So it's a 7.39, it's coming back to normal. So at one point it was an acidosis, but we're within the normal range, so we can call it compensated acidosis. The next thing we're going to look at is the CO2 and the bicarb. Because the pH was low and because the bicarb was low, we can see that it is a compensated metabolic acidosis. CO2 has started to try and pick up a little bit, and add some more or decrease the acid from the situation to try and bring the pH back to normal. Like I said, you'll get a bunch more of these during lab, so if you're so confused, you'll get more. All right, a 24-year-old drug abuser is brought into the ER unconscious. He has shallow breath, looks pale, and is clammy. Blood gases show the following. He's got a low pH of 7.29, a very high CO2 of 50, and a bicarb of 25. And that bicarb of 25 is normal. So the pH is low and the CO2 is high. So for this one, you want to use Rome, respiratory opposite, metabolic equal. pH is low, which means it's acidosis. CO2 is high. One is low and one is high. Respiratory opposite, metabolic equal. It is a respiratory acidosis that's uncompensated. These are the easiest ones to figure out, is when you have that respiratory acidosis, because the pH is low and CO2 is high. And it's usually a patient who's unconscious or hasn't been breathing. All right, which set of results is consistent with an uncompensated metabolic acidosis? So if it's uncompensated, that means that the pH is totally not normal. So if there's a normal pH on there, cross that out. And acidosis means it has a pH of less than 7.4. So now you're down to two choices. If it's metabolic, it would mean that if the pH is low, the bicarb would be high. And if the pH is high, the bicarb would be low. Sorry, if it's respiratory, they're opposite. Metabolic, they're equal. So if the pH is low, then the bicarb is low. Yes, the answer is B. The CO2 is still normal. It's not yet compensated, but the CO2 would come down to compensate over time. Okay, that's our last blood gas. Like I said, I'm not going to go over too many of them because you're going to do a lot of them in your lab this week. So we're going to move on to hemoglobins and porphyrins, and then towards the end of this, we're going to have some review questions going back to things that could be on exam 3 from lipids and things like that as well. Okay. I pixeled screen again. I really fight with this some weeks here. Okay. When it comes to porphyrins and hemoglobin, we're going to start with hemoglobin at the very bare bones of this, and we're going to work our way into what the heck a porphyrin is. So hemoglobin, which you've heard of, heme and globin, is a protein in the body that carries oxygen and CO2 from the tissues to the lungs and the lungs to the tissues and vice versa. It carries it all over the body. It also acts as a major buffering system within the body. So when we do hemoglobin testing, it usually tests for the hemoglobin, which carries the oxygen, which is found inside those red cells. So actually, when we do a hemoglobin test in the lab, the cells are actually lysed during that test. So you can see how much is inside those cells. Hold on a sec. I'm going to pixel screen again. There we go. Here's a picture of what hemoglobin looks like. Now, I know you're kind of in a panic thinking, oh my gosh, do I have to be able to draw that? No, you do not. But you do have to be familiar with this, especially when you get to Hem 1. I think that's next quarter that you have that. In Hem 1, you're going to learn a lot more about the structure of hemoglobin and why it looks like this. There's 2 non-alpha chains, which are sometimes called beta chains, and 2 alpha chains. And it's all held together by this one iron here that you can see in the middle. I'll make it a pretty blue color. So you can see this iron here in the middle. And he kind of holds everybody together. So that's why when somebody's considered anemic, they give them iron pills. And if you've ever been pregnant and taken iron pills, it's very unfortunate. They cause constipation and all kinds of wonderful things. But, yep, that's why they need to take it, to make more of this hemoglobin, which makes more, you need to make more blood cells. Okay, I'm having a hard time switching slides again here. There we go. So the synthesis does occur in the bone marrow. All red blood cells are made in the bone marrow. And we have a couple different types of cells that hemoglobin is made in. 65% is made in the nucleated cells, and 35% is made in the reticulocytes, which are baby red cells. You do need that adequate iron supply, which was that Fe molecule that you saw in there. Then we can also, once we've used up our hemoglobin, we can't just keep making more and more and more and more and more and more and more until we explode. There has to be a way to get rid of extra hemoglobin. There's a couple ways that happens. There's extravascular and intravascular. Extravascular occurs outside the circulatory system within the phagocytic cells of the spleen, which I'm going to show you a picture of in a minute. Also happens in the liver and can happen in the bone marrow. But some of it also happens intravascularly, which is in the bloodstream, where it's just dissociated into alpha and beta dimers, so that's just broken apart just right in the bloodstream. Majority of it does happen in the spleen where it is supposed to happen. And I'm still with you here. I'm just going to get my slide to switch here. Okay, so here's how the synthesis of it does happen. It starts with the combination of glycine aminolevulinic acid synthase plus succinyl-CoA. Those happen inside of a cell, and then you need all of these different things to occur. You need that aminolevulinic acid in the presence of aminolevulinic dehydrase. No, I'm not going to make you memorize all these, which turns into porphobilinogen. You get porphobilinogen deaminase, which goes into hydroxylmethylbilene, which in the presence of uroporphyrinogen 3 synthase makes uroporphyrinogen 3. So you need all of these steps to occur. Now, as you can see, there's all of these enzymes that are necessary for that to happen. We've got this deaminase here, we've got a synthase here, we've got a decarboxylase here, a dehydrase here. So when we talk about those enzymes being necessary for reactions to occur, this is what I'm talking about. All of these steps have to happen. Well, what do you think happens if your body is missing one of these enzymes here? What's going to happen is you're not going to be able to make these steps happen. So if you're missing this uroporphyrinogen decarboxylase, you're going to have a buildup of uroporphyrinogen 3 in your bloodstream, and it's very toxic. But in a normal situation, all of these steps occur, it's very slick, and you end up going to protoporphyrinogen 9, which goes to protoporphyrin 9 with the presence of that little iron molecule that we have here. We end up making heme. Okay, so we got heme here. Oops. Let me circle my heme. And when you add heme with a globin chain, you end up with your hemoglobin, your nice big pretty guy down here. So all of these things need to happen within a cell in order to make hemoglobin. It's amazing that it works as well as it does. All right, so we talked about the synthesis of hemoglobin. Here's the degradation of hemoglobin. So once you've had it, you've used it, it's done its job. You have too much of it, it's not working so hot anymore, your body's able to break it down and get rid of it. So what happens is you got your happy red cell. Here he is, he's floating around the bloodstream. Well, he starts getting a little bit decrepit over time. He starts to get all pycnotic and have like little burrs and things sticking out of him. Well, what happens once he gets old and decrepit is he floats through the spleen, and the reticuloendothelial system within the spleen destroys that cell, and the macrophages, which are the garbage truck of the bloodstream, they're kind of like big white cells, they eat up those red cells. And within that macrophage, okay, and this little square here is a macrophage. It's broken down into iron, globin, and heme oxidase. So the iron is carried by transferrin back to the body where it can be used to make more hemoglobin. The globin part is carried back to the amino acid pool within the liver, because pretty much everything is stored in the liver, right? Then what's left is CO2, or carbon monoxide, or CO2, which is expired through the lungs. You exhale that, so that goes away. And then you've got bilirubin left, okay, which is what our lab is going to be this week. The excess bilirubin is carried by albumin back to the liver, which is where the hepatocyte is. That excess bilirubin becomes conjugated in the liver, where it is then dumped out into the feces and turns into poo. So some of it becomes fecal urobilinogen, which turns into other things, which makes poo brown actually. And then the rest of it ends up back in the bloodstream where it is urinated through the kidneys. So there's two ways of it actually exiting the body. One is through feces and the other one is through urine. So that is exactly what happens to a dead red cell in a nutshell. So what you need to be familiar with, and you're going to get a lot more of this in hematology, is the fact that the spleen kills those red cells, which breaks apart the different pieces, and it is then brought, the bilirubin goes out through the feces and the urine. So you really only need to know that for now. So we do have diseases within that hemoglobin as well. There's all different types of hemoglobin. Now, you don't have to memorize these because your hematology instructor, Lori Bierkley, is going to put you through the ringer with all of these next quarter, I promise. But there are different hemoglobins. There's hemoglobin SC, SC, E, and D, plus probably many more out there. So different types of hemoglobins have different functions within the body. Now, if you have an absence of one of these, or you have too much of another because of a genetic defect, it can cause a lot of problems. Some of you may have heard of sickle cell anemia by Kathy. Sickle cell anemia causes a lot of problems with the red cells and the structure of the red cells. So once there's not enough hemoglobin, the cell starts to sickle and twist, and it doesn't go through the bloodstream very easily, and it doesn't carry oxygen very well at all. So what we do to test for that can be something called electrophoresis. And I have a picture of a patient down here at the bottom. Now the top person here is the patient. Okay. So the patient here, what we do is we add some of the patient sample at the beginning, and I'll put a little spot here as to where the patient may have been added. Okay. So they put the patient on there and they expose it to a electrophoresis card. So they travel from slowest to fastest on this card when you expose it to electricity. So they travel at a rate from fastest to slowest, where at a pH of 8.6 is CSFA. And here, let me do this here. You would put the patient on the other side. Okay, so here's where our patient started. So the slowest one to travel is C, and then S, and then A. So C only travels so far. So this person down at the bottom has C, S, and A present. The next person, which is a normal patient, has a lot of A present. This next person here looks like they've got a lot of A and some S. So they have a sickle cell trait. This person at the top, as you can see, because this whole row here is all hemoglobin S, this person, all of their hemoglobin stopped at the S stage. Okay? So this person on the very top has a lot of S, so it's a sickle cell patient. But it's important to know that the migration from slowest to fastest on cellulose acetate at a pH of 8.6 is CSFA. That's the order that it goes in. One of the other disorders that you can have is called a thalassemia. A thalassemia is a decreased production of alpha or beta chains. You're going to learn all about these in hematology as well. So not just having too much of hemoglobin S and things like that can be a problem, but those alpha and beta chains that those hemoglobin molecules made out of, if you have a decrease in one or the other of those, you have what's called a thalassemia. They do have some crazy names, and you'll learn about that in hematology. But just for the scope of this course, know that decreased production of alpha or beta chains is a thalassemia. Some of the ways that we test for it, we talked about the citrate agar electrophoresis here that I mentioned on the bottom. He's on here. But also a solubility test is what we do for the hemoglobin S or the sickle cell. And we can do the cellulose acetate, hemoglobin electrophoresis is what I mentioned on the last slide. There's also citrate agar electrophoresis. So those are the 3 ways that we can test for abnormal hemoglobin. All right, let's talk about these porphyrins and porphyrias. You can see this poor guy down in the bottom right-hand corner. A lot of people see that and go, oh my gosh. He has something called Porphyria cutanea tarda, I believe. I'm not 100% sure, but by the looks of his skin, I'm thinking that would be the one that he has, and we'll get to that in a little bit. The main purpose of a porphyrin is to combine with iron to make heme. And if you are missing any of those enzymes, you end up, that I showed you earlier, you end up with an accumulation of those heme precursors, like the uroporphyrinogen and the copoporphyrinogen, all of those ones that I mentioned earlier. So if the enzyme is missing, one of those will build up. When they build up, we find them in the urine and in the feces and in the blood as well. And we'll get to in a minute why that guy looks like he does. Okay. So here's a little slide that I found that's kind of fun. It kind of goes along with the cartoons like we used for the blood gases last week. And I'm not going to make you memorize these heme precursors, but Kevin probably will. Kevin is our lab adjunct. Lori probably will. The Porphyrin synthesis, in order to memorize it, here's a little cartoon note to make it easier. Remember I told you that delta aminolevulinic acid went to porphobilinogen, went to uroporphyrinogen, went to copoporphyrinogen, went to protoporphyrinogen, which gives you heme, plus a globin gives you hemoglobin. So if you're missing any of the enzymes, which would be where the arrows are, that's where you would have something build up. So if you want to memorize the flow of what these are, want to pour your pronto a cup of heme. So if you remember that saying, you'd be able to know what the order is of these. So once again, a porphyria is the excess formation of one of those porphyrins. So for example, it would be a buildup of this, or it could be a buildup of any of these, because you're missing an enzyme. So here's another example. Heme is a substance that we find in all of the body's tissues. Well, when we have an excess of the porphyrin, it causes photosensitivity. So if you've got a buildup of those porphyrins in your blood, it causes photosensitivity in your skin. So you can see this guy's neck down here. And when the porphyrins are exposed to light and oxygen, they generate a charged, unstable form of oxygen that damages the skin. You can end up with nerve damage, pain, paralysis. Sometimes it's so severe that it leads to respiratory paralysis as well, where they're unable to speak or swallow. It can be very painful. Of the porphyrias that I have seen in the hospital and have drawn blood for, they've all come in with this type of photosensitivity, which leads the doctor to wonder if that's what the problem is. They usually, their arms, anything that's exposed to the sun gets really red and almost blistered. Underneath their clothes, their skin looks fine. So that kind of tips them off a little bit. So these porphyrins, they're relatively stable. They do fluoresce red when excited by light near 400 nanometers, so they do have special lights and things they can use to look at the skin. There's 3 that are clinically significant that you need to be familiar with. Protoporphyrin is the one that shows up more in the feces if you have an excess of that. Protoporphyrin and poop both start with a P. Okay, so that one's easy to memorize that way. Uroporphyrinogen is found in the urine. So uroporphyrinogen starts with a U, so does urine. Copoporphyrinogen can be found in both. So co meaning two, like a co-partner, so it's found in either one. If we have an excess of any of these porphyrins, it indicates abnormal heme synthesis, and it can cause a problem. The porphyrinogens are reduced functional form. They don't fluoresce and don't cause as much problem. So the disease correlation, what we're looking for, the problem is caused by an enzyme deficiency. You're missing an enzyme to cause those steps to happen that we saw earlier. When we have a disease, we call it a porphyria. So that is an inherited or acquired enzyme deficiency that results in the overproduction of a certain heme precursor because it can't be broken down to the next one. There's a couple different types, and these are just for your information. You don't need to memorize these, but there's some that cause neuropsychiatric symptoms, so you would have like brain issues with it. If you're missing aminolevulinic acid dehydratase, if you have a deficiency porphyria or an acute intermittent porphyria, those can cause psychiatric problems. Like I said, don't memorize these. This is just interesting. Some of them cause cutaneous symptoms or the skin symptoms, and these are the ones that I have seen of the very few actual porphyria patients I've seen in my life. Most of them had the porphyria cutanea tarda. There's also hepatoerythropoietic porphyria, which involves the liver and the red cells. You can have just an erythropoietic porphyria, which affects mostly the red cells. And you can have a congenital erythropoietic porphyria, which means you're born with having a red cell production problem. There can have neurocutaneous, which means that you have it in your skin and in your brain, which would be hereditary coproporphyria, and a variegate porphyria. So like I said, you don't really need to memorize all these for the scope of this course. Be familiar with what a porphyria is, what it means when they have a porphyria, and you could be familiar with the fact that porphyria cutanea tarda is the most common. Keep that in mind when you do your discussion post for this week too. All right, so how the heck do we test for these things? Not something we're going to do in this class, but we're definitely going to talk about it in your urinalysis class coming up. We do test urine for these. Urine is a very easy test to do, very non-invasive. We use what's called the Watson-Schwartz or the HESH test. These are great board questions. So you're looking for someone with one of these disorders, Watson-Schwartz or a HESH test. For porphyrins in the blood, or in the skin, we can use a Woods lamp, which you've used in biology or A&P, where you looked on the skin for it to glow. So you would notice that those porphyrins would glow at 400 nanometers. There's also the chromatographic separation and quantitation using spectrophotometry and fluorometry and molecular diagnostics. So that gets into a really heavy detailed type of instrumentation that would be needed for those. All right, so why are we doing bilirubin in lab this week? Well, because when we have the breakdown of hemoglobin from RBCs, we end up with the unconjugated bilirubin, which floats around after it gets broken apart in the spleen. From there, that unconjugated bilirubin heads to the liver, where it then becomes conjugated by something called bilirubin diglycuronide. So those combine together and end up in the intestine, where we've got fecal urobilinogen, which turns to fecal urobilin in the poop and makes it brown, or some of it heads to the kidneys where it's excreted as urine urobilinogen. So bilirubin does play a part in the whole process of the breakdown of hemoglobin. So we threw bilirubin in with this week's laboratory. All right, Victoria, you're my only person here, so I'll try and play along with you for some of these questions. All right, so I'll answer too, so nobody knows who's getting what. I'll get them wrong. Which fatty acid does HDL carry back to the liver? Okay, the answer for this one is cholesterol. Cholesterol does carry HDL back to the liver, which is why HDL is such a beautiful thing. If you don't have enough HDL, you can have excess cholesterol build up in your bloodstream, which can help lead to some of that, the plaque and atherosclerosis and things. All right. Which of the following provides a rich source of energy, are an integral part of cell membranes, and play a structural role in cells? Yep, the answer is lipids. Well, protein seems like it would be a good answer because there are a lot of proteins within your red cells. Lipids actually are the rich energy source that play an integral part of cell membranes. It's what keeps them from touching each other and keeps them floating around because they've got the polar heads and tails and things. All right, what is the difference between a phospholipid and a triglyceride? Phospholipids have a phosphate group instead of a third fatty acid. Phospholipids don't even have fatty acids. Phospholipids contain 3 phosphate groups, and phospholipids are hydrophobic and water insoluble. Okay, the answer for this one is phospholipids have a phosphate group instead of a third fatty acid. Which of the following is responsible for the creamy look of a lipemic specimen? The answer for this one is chylomicrons. Chylomicrons are what make it look creamy. Okay, how long should you fast before having a lipid panel drawing? Okay, the answer for this one is 12 hours, actually. You should wait 12 hours. So most people have their lipids drawn at in the morning, so they just don't eat after dinner time at night. Which of the following are amphipathic? Yep, the answer for this one is phospholipids. Phospholipids are the ones that are amphipathic. Okay, one more question. If your triglyceride is greater than blank, you cannot calculate and do the LDL using the Friedewald calculation. Okay, the answer for this one is 400 milligrams per deciliter. If your triglyceride is higher than that, you cannot calculate the LDL. You can't even use that calculation. You'd have to have the LDL done using beta modification or some other type of testing technique. The calculation would not be accurate. Okay, so the one last thing I want to mention for your exam 3 is make sure that you know how to calculate the following. That you know how to do a, I'll put these up here, calculate the Friedewald for LDL. You'll have some of those on your test. Oops, that was supposed to say LDL. And you're supposed to be able to know how to do an anion gap, know what it means if it's normal or abnormal. You have to be able to calculate an osmolality, and you also have to be able to do an osmolar gap. I can absolutely promise you with 100% that those things are on your test. You'll need a calculator to calculate those. You cannot use any notes. You have to be able to do a couple Friedewald calculations, a couple anion gaps, a couple osmolalities, and an osmolar gap. So if you go to the test and say, oh, I didn't know I had to do those, I'm telling you right now that you do have to be able to calculate those, so know those calculations. It's probably the last 10 questions on the test. So out of 50 questions, that's about 20% of your grade. If you get all those wrong, that drops you down to an 80%. Make sure you know how to calculate those. It's open-ended, not multiple choice. Okay, any questions? I'm trying to change that to no. No? Okay, sounds good. I'm going to stop the archive then.